And dear students of 12th class, today in your botany we are going to take up the salient features of the double helix structure of DNA. And previously we have talked about the structure of the DNA. Most probably you must be remembering, and this being the most important topic in this what we are studying, that is the molecular basis of inheritance. And from the examination point of view, this is going to be the most important chapter, that is the structure of DNA. And as you must be knowing that this DNA now is having a double helix structure as proposed by Watson and Crick. This was the ultimate structure about the ultimate truth about the structure of the DNA. After so many chaos and so many researches, it was ultimately decided that the DNA molecule has having a three-dimensional double helical structure there with two strands internally which are connected through the base pairs. Bases are in the form of adenine and thymine. Adenine is connected to thymine, whereas guanine to cytosine. That is a purine base against a pyrimidine in the double helical structure, and likewise the guanine against the cytosine. Adenine against thymine and guanine against cytosine. Adenine and guanine, the purines, and thymine and cytosine, the pyrimidines. And this is in case of the DNA, while as in the RNA, you must be knowing that in place of thymine, there exists the uracil as the base, as the nitrogen base. That means thymine is replaced by uracil. And this is the open structure of this double-stranded polynucleotide chain. You can visualize here a pentose sugar and the phosphate moiety that forms the backbone of the strand or the DNA, and in between the two strands are connected through the nitrogenous bases. Adenine with thymine, guanine with cytosine. Adenine thymine connected through double hydrogen bonds, whereas guanine and cytosine by a triple hydrogen bond. Always the two strands, they'll run anti-parallel to each other. Now that will come in our discussion about the salient features of this double helix model of DNA. The salient features of the double helix structure of the DNA are: it is made up of two polynucleotide chains, where the backbone is constituted by sugar phosphate. Sugar phosphate constitutes the backbone of this polynucleotide chain, and the bases, nitrogenous bases, they project inside. And the two chains have anti-parallel polarity. It means if one chain has the polarity 5-3' and the other has 3' and 5'. The bases in two strands are paired through hydrogen bonds, forming base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand, and vice versa. That's guanine with cytosine. That is, and similarly, guanine is bonded with cytosine with three hydrogen bonds. This results that a purine comes opposite to a pyrimidine, and this generates approximately uniform distance between the two strands of the helix. and the two strands are coiled in a right-handed fashion. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of a meter, that is 10 raised to the power minus 9 meter. And there are roughly 10 base pairs in each turn. 10 base pairs in each turn. Consequently, the distance between a base pair in a helix is approximately equal to 0.34 nanometer. The plan of one base pair takes over the other in double helix. This in addition to hydrogen bonds confers stability of the helical structure. And now compare the structure of the purines and the pyrimidines. This is here purine versus a pyrimidine, adenine, thymine, and guanine, cytosine. You can find the distance between the two polynuclear chains in the DNA remains almost constant. The proportion of a double helix structure for DNA and its simplicity in explaining the genetic implication became a revolutionary model proposed by the Francis Crick and James Watson, and they ultimately gave very soon. Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA, then to RNA, and then ultimately expresses in the form of protein. And this is here the central dogma. DNA has the capability of getting replicated, that produces its own replica. Then after transcription, it gets transferred to messenger RNA, and then the message gets translated to form the proteins. That is the ultimate product of this all. Proteins are the ultimate, they which are expressive in nature. And this, well as in some viruses, the flow of the information is reversed, because these viruses are always somewhat, these are unpredictable creatures, that is from RNA to DNA. In some viruses, the information is in the reverse direction, that is from RNA to DNA. And this is the central dogma. The information gets transferred from DNA through messenger RNA, and then ultimately to proteins. And that is all for today. Thanks. Have a nice day.